Okay, ladies and gentlemen, you are welcome. So in today's lecture, we are going to look at pentose phosphate pathway. Introduction to pentose phosphate pathway. The pentose phosphate pathway is a metabolic pathway parallel to glycolysis. And why is it that it is called parallel to glycolysis is because pentose phosphate pathway and glycolysis share similar substrates. Remember, glucose is converted into glucose 6-phosphates in glycolysis, and the glucose 6-phosphate will continue into the glycolytic pathway. But at the same time, the glucose 6-phosphate also serves as a substrate to pentose phosphate pathway. So that is why pentose phosphate pathway, because they are sharing the same substrate, is considered as a parallel pathway to glycolysis. And the primary functions of pentose phosphate pathway, at the end of the pathway, it generates NADH and pentose phosphate pathway, and pentose, which are necessary for cellular processes. So in pentose phosphate pathway, NADPH and pentoses are generated, and these two important molecules are very essential for cellular processes, including prevention of oxidative stress, production biosynthesis, the DNA replications. All these are cellular processes that require the presence of NADPH and pentoses. So the pathway is essential for cellular redox balance and biosynthesis. The NADH and the pentoses, they are very important for redox balance because in most cases, provided that we take air, that we respire, a lot of oxidatives or oxidants are generated. So it is the work of the NADH to neutralize the stress caused by this oxidant. So that is why the pathway is important for redox balance due to the presence of NADH generated, as well as the biosynthesis of other important molecules in our body. As you can see, this is the starting point of the pathway. That is why the pentose phosphate pathway is considered as a parallel to glycolysis. As you can see from here, ladies and gentlemen, clearly that at the first step, glucose 6-phosphate is the first substrate to start pentose phosphate pathway, which would be converted into glucose, which will be converted into 6-phosphogluconolactone. And then it continues like that. So, as you can see, that ladies and gentlemen, the overview of the pathway. So the pentose phosphate pathway consists of two main phases. We have the oxidative phase and we have non-oxidative phase. So these are the two phases of PPP, that is pentose phosphate pathway. And this pathway, it primarily takes place in the cytoplasm of the cells. The pathway provides key intermediate for nucleotides and amino acid biosynthesis. Why? Because at the end, ladies and gentlemen, as you can see from here, this is the pathway where we have glucose 6-phosphate is converted into 6-phosphogluconolactone. So this pathway that we are demonstrating is an example of oxidative phase, where at the end, the glucose 6-phosphate is converted into ribulose 5-phosphates. And as we can see from the steps, we have first glucose 6-phosphate is converted into 6-phosphogluconolactone, and then we have 6-phosphogluconates, which converted from, or which is generated from 6-phosphogluconolactone using an enzyme called lactonase. And then the enzymes called 6-phosphogluconate dehydrogenase convert 6-phosphogluconate to ribulose 5-phosphates. And of course, ladies and gentlemen, this ribulose 5-phosphate will be converted into ribose sugar. And of course, if you can remember, ladies and gentlemen, ribose sugar is one of the sugars, or is one of the major component of nucleotide. Remember, the nucleotide are formed due to combination of phosphate group, the ribose sugar, and the nitrogenous bases. So that is why the pathway it provides a key intermediate in nucleotides and also amino acid synthesis, because at the end of the oxidative phase, ribulose 5-phosphate is generated, and also 2 molecules of NADPH, as we can see, are generated in the oxidative phase. So now let's look at the oxidative phase details. So during the oxidative phase, glucose 6-phosphate is converted into ribulose 5-phosphate. So it started with the glucose, and the glucose 6-phosphate is converted into ribose 5-phosphate. So this phase produces NADPH, and this NADPH is very important in reductive biosynthesis and antioxidant defense. Remember, ladies and gentlemen, oxidative stress is a stress caused by oxidant, and these oxidants are the chemical species that are produced in the body, and they are always looking for the electron-rich center to attack and destabilize them. So one of the very important role of this NADPH generated, it serves as an antioxidant in order to prevent the oxidative stress. So this is the pathway. As we started the discussion, we have 3 interchangeable phosphate pentoses. So as we can see, ladies and gentlemen, we have glucose 6-phosphate, which converted into 6-phosphoglucanolactone, which converted using an enzyme glucose 6-phosphate dehydrogenase. And in this stage, as we can see, 1 molecules of NADPH is generated. And then from 6-phosphoglucanolactone, lactone is converted into 6-phosphogluconate using an enzyme called gluconolactonase or lactonase enzyme. And then, of course, the 6-phosphogluconate is converted into ribulose 5-phosphate with the removal of 1 molecule of CO2 from the 6-phosphogluconolactone. And the enzyme that do this work is 6-phosphogluconate dehydrogenase enzyme. And as you can see, another molecules of NADPH is generated. So that is what makes the total of 2 NADPH in the pathway that are generated. Then the enzymes, which is glucose 6-phosphate dehydrogenase, catalyzes the first, and also is considered as a rate limiting step. That is the control point of the pathway. So this is a point of regulation in oxidative phase or pentose phosphate pathway. So the next one is non-oxidative phase details. So in the non-oxidative phase, it involves reversible interconversion of sugar phosphate, and it allows the cell to generate ribose 5-phosphate or regenerate glucose 6-phosphate. So transketolase and transaldolase are the enzymes that are enzymes facilitating this reaction. So you see at the end, the non-oxidative pathways, it helps the cell to generate ribose 5-phosphates, and also it regenerates another glucose 6-phosphate for the continuation of either the pentose phosphate pathway or glycolysis. So let's look at the pathway very clear. So you see, the first thing, we have a step where ribulose 5-phosphates are generated in the oxidative phase. So that ribulose 5-phosphate, it will be converted into ribose 5-phosphates, and then this ribose 5-phosphate will now condense itself with xylulose 5-phosphate to generate ceduloheptulose 7-phosphate with glyceraldehyde 3-phosphate using an enzyme called transketolase. The transketolase is an enzyme that convert the 2 molecules that convert ribose 5-phosphate with xylulose 5-phosphate to generate ceduloheptulose 7-phosphate and glyceraldehyde 3-phosphate. And then another enzyme called transaldolase will now convert the cedulloheptolase 7-phosphate and glyceraldehyde 3-phosphate into fructose 6-phosphate and erythrose 4-phosphate. And then, of course, these fructose 6-phosphates can be isomerized to glucose 6-phosphate, as we can see from here. And this glucose 6-phosphate can be used to continue the pentose phosphate pathway, or will be channeled for glycolysis or other important metabolic pathways. Then at the same time, the erythrose 4-phosphate will now condense with another molecules of xylulose 5-phosphate using transketolase enzymes to produce another molecules of glyceraldehyde 3-phosphate and fructose 6-phosphate. And of course, the fructose 6-phosphate, through gluconeogenesis, it will now convert into glucose 6-phosphate. And then also at the same time, the glyceraldehyde 3-phosphate generated here, it will also undergo gluconeogenesis to generate another molecules of glucose 6-phosphate. So these are the important biomolecules that are generated in the non-oxidative phase. We generate glucose 6-phosphate, and we also regenerate ribose 5-phosphate. So as we can see, ribose 5-phosphate is generated, and then glucose 6-phosphate are also generated. So then the next thing, what is the role of NADPH in a cell? So NADPH produced in the pentose phosphate supplies reducing power for fatty acid and nucleotide biosynthesis or synthesis. So this NADPH is very important in fatty acid and nucleotide biosynthesis. It also plays a critical role in maintaining a reduced state of glutathione. Glutathione is an antioxidant molecules that used to prevent oxidative stress. So usually this glutathione is converted into oxidized form when it donates an electron to oxidant using an enzyme glutathione reductase. So when you have the oxidized glutathione, so it means that the electrons is already lost by the molecule. So therefore, for it to continue with donating more electrons, or for it to maintain its antioxidant property, so it needs to be in a reduced form. That is, as it donates electrons, so it needs to accept more electrons for it to be in a reduced form, so that whenever there are other oxidants present in a cell, it will continue neutralizing them. So what happened? It is this NADPH that usually donates electrons to glutathione, thereby maintaining its reduced form. Therefore, as a result of this, maintaining the reduced state of glutathione is important in combating oxidative stress. So cells with high anabolic activity or oxidative stress depend heavily on NADPH supply. And if there is no heavy supply of NADPH, so there is going to be oxidative stress. So what are other roles of NADPH? As we can see, it's used in reductive biosynthesis. It's also very important in detoxification of toxic substance. Cytochrome P450 is an enzyme that is very important in preventing detoxification in the body. So it helps in detoxifying foreign substances that are toxic to the body. Then it's also very important in as an oxidative defense, and also it helps in ROS generation. So this is actually some of the 4 important role of NADPH in a cell. So what are the biological significance of the pathway? The pentose phosphate pathway is rapidly dividing cells, such as cancer cells, for nucleotide synthesis. So in cancer cells need a constant division. So therefore, for it to divide, it requires or it needs the NADPH for nucleotide biosynthesis, as well as the ribose sugar generated in the pathway. So that's why sometimes if you really want to cure cancer or develop cancer drugs, you can decide to target pentose phosphate pathway so that the ribose sugar as well as NADPH will not be there. So therefore, there will be no reductive biosynthesis. And also the pathway, it provides a metabolic flexibility, balancing the need for NADPH and nucleotide precursor. So the pathway also supports the detoxification process in cells by supplying NADPH. So what are the regulation of the pathway? What are the regulation of the pentose phosphate pathway? Glucose 6-phosphate dehydrogenase is the primary regulatory enzymes of pentose phosphate pathway, and the activity of this glucose 6-phosphate is regulated by availability of NADPH and cellular demand for NADPH. So high amount of NADPH in a cell blocks glucose 6-phosphate dehydrogenase, while high amount of NADP plus activates the glucose 6-phosphate dehydrogenase enzyme. Therefore, the feedback inhibition by NADPH helps to maintain cellular redox hemostasis. So NADPH is a final product of the pentose phosphate pathway, that is oxidative phase. So remember, when you said feedback regulation is when the final product of a pathway inhibits the first enzymes of the pathway. That is what you call feedback regulation. So as we can see, this is the regulatory point where we have the glucose 6-phosphate dehydrogenase. So the pentose phosphate pathway, this is the point of regulation. So glucose 6-phosphate dehydrogenase is the rate limiting enzyme and is controlled by, there are two ways that you can regulate this enzyme. So you can regulate it using allosteric regulation. So allosteric regulation, at this point, you have a feedback regulation where the NADPH, which is the end product of the pathway, regulates the pathway by binding the NADPH to allosteric side of the enzyme. Then we have inducible enzymes. The synthesis of glucose 6-phosphate dehydrogenase is induced by increased insulin or glucagon ratio after a high carbohydrate. So the glucose 6-phosphate dehydrogenase enzymes is a protein, so therefore it's a gene. It is one gene, so the synthesis or the expression of this glucose 6-phosphate, it depends on the insulin and glucagon ratio. So if you have high insulin, lower glucagon, the pathway will be activated. And we have high glucagon, low insulin, the pathway will be blocked because of inhibiting or downregulating the expression of the glucose 6-phosphate dehydrogenase gene. So what are the clinical relevance of this pathway? Deficiencies in glucose 6-phosphate can lead to hemolytic anemia, especially under oxidative stress, and the pathway's activities is altered in certain cancer cells, highlighting its role in cell proliferation. So drugs targeting pentose phosphate pathways are being explored as potential treatment for metabolic disorders. So ladies and gentlemen, this is where we are going to stop. Please, if you know that this is the first time you come to my YouTube channel, please subscribe and join the YouTube membership. See you next.